Hi, I'm Daniel, a member of the prestigious Group Number 9 of Georgia Tech's Biology 1520 class. I'm here today with Hope, Lien, and Alex to talk to you guys about some research that has come out of labs at UC Davis involving optogenetics. An article from Scientific American described optogenetics as the combination of genetics and optics to control well-defined events within specific cells of living tissue. What makes this nascent technique so promising is how scientists are able to precisely stimulate specific neurons via light and see how they respond to inputs. One application of optogenetics was shown in an experiment out of MIT to study blindness. The techniques they use to study blindness are similar to how they study other functions or malfunctions of the brain. As you can see in the diagram, their experiment began by taking a piece of DNA from algae that codes for proteins called channel rhodopsins. These proteins are normally found in the eye and respond to light by depolarizing the cell. In this experiment, they took that gene and placed it directly in mice's neurons. These neurons would respond to light by sending an action potential, which allowed mice to respond to light without requiring input from their eyes. The mice that were treated with this gene were able to follow a maze, which had an illuminated exit, as well as mice that could see normally, and much better than mice that could not see. Optogenetics can be used to study how many of the brain's functions can be affected by light. Next, Lien is going to talk about how the brain stores short-term memories.
Memory is stored in two ways. The first way includes your short-term memory, and the second mechanism is storage in your long-term memory. Let's begin with discussing the process of storing short-term memories, which takes place in the hippocampus. The hippocampus is the paired horseshoe structure in the brain that is part of the limbic system. Its main purpose includes memory formation, organization, and storage. As we have learned in class, it activates PKA, protein kinase A, which activates ion channels and proteins in the presynaptic cell. For the short term, the communication between these cells in the hippocampus is enhanced. Think of short-term memory like your brain's post-it note. It can only hold a few items and can easily be lost or forgotten if not put into a permanent place. On the other hand, long-term memory includes the synthesis of new proteins. As a result, we see the creation of new synaptic pathways strengthen the communication between cells in the cerebral cortex. Hope is going to tell you more about an optogenetics experiment affecting the recall of short-term memory in mice.
In a recent experiment, Kazuma Tanaka, Brian Wilkin, and other researchers at UC Davis Center for Neuroscience and Department of Psychology used light to block memory retrieval pathways in mice. They first trained the mice to have a fear response by shocking them when they were placed in a cage. This caused the mice to freeze in place instead of moving around. Tanaka and Wilkin identified the nerve cells involved in learning and memory recall located in the hippocampus. Wilkin said, "The theory is that learning involves processing in the cortex, and the hippocampus reproduces this pattern of activity during retrieval, allowing you to re-experience the event." Using optogenetics, the researchers were able to switch off these specific nerve cells, which prevented the mice's retrieval of the memory of getting shocked, and the mice explored the cage instead of showing the fear response. Tanaka and Wilkin also showed that when other cells in the hippocampus were turned off, the mice's memory recall was not affected. And here's Alex as to why this experiment is important.
In conclusion, optogenetics demonstrates that neurons can be genetically modified to be controlled by light. For the mice of this particular experiment, exposure to light inhibits the retrieval of a memory. So why is this important? This experiment and other studies involving optogenetics are improving our understanding of the human brain and how its neurons interact to initiate action or thought. As more research is conducted, the goal of developing a method of therapy for human brain disorders becomes more and more attainable.